Hello, my name is Chiara Tontini. I'm a research associate at the University of Manchester, and on behalf of my co-authors, I'm excited to present you a study recently published in Allergy, focusing on mast cells, omics, and AI. Mast cells are tissue sentinels that constantly sense and adapt to their surrounding microenvironment. This incredible ability to adapt, also called plasticity, is vital to respond to threats, but it's also a huge challenge to study. In fact, we don't have many tools to dissect plasticity in tissues, making it very difficult to predict what mast cells will do, especially in disease where many inflammatory signals like cytokines can profoundly influence mast cell responses. So, how do we try to solve this? We decided to use primary human mast cells and expose them to different cytokines mimicking inflammation, tissue damage, allergy, and profibrotic environments for 24 hours. We then extensively studied their effect on our in vitro model, including changes in gene expression, surface receptors, and functional assays. We then used machine learning to identify interesting genes to create minimal cytokine-activated signatures and use them to reanalyze publicly available skin and respiratory single-cell datasets. By using transcriptomics, we were able to characterize both unique and shared genes expressed following different cytokine stimulations. By using functional assays, we measured the positive or negative effects of cytokine priming on both the granulation and cytokine release, demonstrating how profoundly cytokines like interferon gamma, TGF-beta, IL-33, and IL-4 and IL-13 can shape mast cell activation. Using our machine learning approach, we successfully reduced the number of genes we needed to analyze to just 245 in total. This simplified set was still incredibly accurate at identifying cytokine priming. By applying these signatures to public datasets from skin, nose, and lung tissue, we could identify cytokine priming in mast cells from different diseases like atopic dermatitis, COPD, COVID-19, and pulmonary fibrosis. This also allowed us to define which disease-preferred activation trajectories mast cells tend to adopt. This is just a glimpse of what we've done, and there is so much more to discover in the full article. If you found this work interesting, I highly recommend you following the links below. A huge thank you to all my co-authors, our collaborators, and our funders, and thank you for watching.